All right, so hello everyone. Welcome to this time's Zoom seminar. I just want to make a special shout out. Roxane is my hero. She saved the day. Marco, who was originally planned to give us talk, unfortunately has some injury. But hopefully he will feel better soon and then maybe come back later, give us another lecture. But Roxane is really my hero. So thank you so much, Roxane. So it's really my great pleasure to welcome today's speaker, Dr. Roxane Tussiwand from NIDCR, who's my also very close colleague and friend. So Roxane currently is Statman investigator in Immune Regulation Unit in National Institute of Dental and Cranial Facial Research, NIDCR. So she graduated with a biology degree from University of Milan in Italy, where she studied childhood leukemia. And then 2006, Roxane was sponsored by the Italian government to pursue her PhD training in molecular medicine at the Institute for Research in Biomedicine in Bellinzona, Switzerland, where she studied dendritic cells development, which I believe we're going to hear a lot today. Following a postdoc training at the University of Basel, Roxane then joined Ken Murphy's group at WashU, St. Louis, where she studied transcriptional regulation of dendritic cell development. And from 2014, Roxane actually was appointed as assistant professor at the University of Basel in Switzerland. But then she made a really bold move from 2019. Roxane moved her lab from Switzerland to US, joined NIH as Statman investigator in NIDCR. So the research work in Roxane's lab mainly focused on understanding how transcription factors modulate the lineage differentiation and function of immune subsets, which including a couple different directions, including first to study development of hematopoietic cell and their transcriptional network, which promotes the progression from a uncommitted self-renewing stem cell progenitor to a mature cell. This actually, they try to understand the genomic landscape as well as the transcriptional profile of each subset and their functional properties. Second part is focused on the dendritic cell, as I just mentioned. It's also been a deep love for Roxane's lab. She actually studied the different dendritic cell subset and then how they develop and then how they function under steady state as well as under inflammatory conditions. Now, last part is our lab study early hematopoietic lineage commitment to understand how lymph node and then myeloid commitment is transcriptionally defined, and then whether the lineage of origin translated into specialized functional properties across different subsets. So I think lots of us actually read and studied a lot of Roxane's work from a lot of top tier journals. And with all these achievements, Roxane was awarded by numerous awards and honors, which including 2014 Swiss National Science Foundation professorship, 2016 Thornback Award Society for Leukocyte Biology, and then 2018 she was appointed as Earl Statman tenure track investigator in NIH. So we all very excited here. Roxane, I think this is the second time I hear your talk. Please go ahead. Thank you.
Yeah, thank you so much. It's a pleasure to be here, and it's a pleasure to be in front of like a large audience that you're organizing, which is connected to China, as I just got back 2 days ago from China, and it was such a pleasure to be there and to meet with colleagues and friends now. And so I'm really looking forward to establish more collaborations and networks. So what I'm going to talk today is like about plasmacytoid dendritic cells and how they are close cousins of dendritic cells. Okay, so but first, let me introduce the team, which is the people who do the work And in particular, the talk I'm going to talk today is the work of a past PhD student, Patrick Rodriguez, who is now in Marcos' lab. So I thought that would be the closest to Marcos' lab I could do. Okay, so, but let me start with introducing the dendritic cell subset. And dendritic cells are highly specialized, and I'm trying here to simplify the number of subsets, but this number with the availability of single cell technology has massively increased. However, we can identify in particular type 1 dendritic cells and type 2 dendritic cells, besides plasmacytoid disease and monocyte-derived disease. And there is still a lot of work which is being done around the other two subsets, but let's focus on PDCs. So PDCs are known for their antiviral properties, which is linked to their capacity to recognize viral particles and produce high amounts of type 1 interference. On the other side, PDCs were more controversially linked to the antigen presentation capacity, with labs showing that they can do it, labs showing that they cannot do it. What is clear is that there must be, or has been suggested, that there is a link with autoimmunity, and in particular with autoimmunities which are characterized by high amounts of type 1 interferon, such as lupus or Sjögren's syndrome. And they have recently been used in a clinical setting for anti-tumor properties. Let me move maybe this to the other side. And the data has been recently released with mixed capacity or reduced capacity as compared to myeloid disease in terms of anti-tumor immunity. So while this side of, or these functional properties of PDCs were very well characterized and accepted, all this antigen presentation, their involvement in anti-immunity, remain controversial. And we decided to focus our attention on the subset. The way we study dendritic cells is really like looking at their development and trying to understand from their development what their properties are. And what was shown already in 2013 by Ken Shortman was that PDCs were capable of differentiating from myeloid progenitor, which give rise to all the other conventional type dendritic cells, and expresses the macrophage colony stimulating factor receptor of CD115, but also from lymphoid progenitors which express the IL-7 receptor or CD127. The transcription factor had been pretty much established throughout the years, and more recently ZEB2 was recently identified as important for PDC development. But one of the key transcription factor is definitely E22 or TCF4. So in order to understand their functional properties, we went back, as I said, to their development. And so what we did, we sorted within the stem cell compartment, which is characterized by FLT3 and by intermediate to low expression of c-KIT, we identified the lymphoid progenitor, which express the IL-7 receptor, the myeloid progenitor or common DC progenitor, which express the CD115, and we included in our study also the double negative population, which was suggested by the group of Oteki in Japan, and in particular Nobuyuki Onai, to be a subset which was enriched with plasmacytoid precursors. And we compared their capacity of these progenitors to generate plasmacytoid dendritic cells in vitro as well as in vivo. And so we put them, so when we put them in FLT3 ligand culture, which allows for the differentiation of PDCs or promotes is instructive for dendritic cells and plasmacytoid development, what we can see is that the percentage of PDCs in terms of output of this culture is the highest when we use the IL-7 or the lymphoid progenitor, suggesting that the highest precursor frequency for PDC is within this compartment. And this compartment is the only one which will differentiate into pro-B cells, which express CD19, while there is no lymphoid precursor potential within either the CDP or the double negative progenitor. So we then put all this subset in competitive setting to understand whether the capacity of the different progenitors was related to the individual culture and whether in competitive setting one or the other subset would be outperforming. And what we saw is that independently of which precursor we used, whether the double negative or the myeloid progenitors, the lymphoid precursor were the best in terms of generating PDCs. When we did that in vivo, upon in vivo transfer, what we saw is again the same thing. IL-7 receptor positive progenitors were the cells which had the highest precursor potential to generate plasmacytoid dendritic cells, not only in vitro, but also in vivo. While when we looked at conventional dendritic cells, those were mostly generated from the myeloid progenitor or common DC progenitor, as it was shown before by many groups, including Nussenzweig and Schalin-Naik. So what was based on this data, we decided to look more into the commitment, because these IL-7 receptor positive cells were capable of generating B cells or pro-B cells, but also PDCs. And so we decided to use a combination of two other markers, Ly6D and Siglec-H. Ly6D, which was shown to be expressed by PDC progenitor from the data generated using the Siglec-H DTR mouse. And we compared, again, these three subsets towards each other. And what we observed was that the double positive for Ly6D and Siglec-H was exclusively committed to generate PDCs, suggesting that the cells or that this precursor was committed to the plasmacytoid lineage. And indeed, the B cell potential was lost within this progenitor when the cells were cultured in the presence of OP9 stromal cells, which and the B cell potential was mostly confined within the Ly6D single positive progenitor. So when we cultured the cells, it took about 4 days, 3 to 4 days to generate mature plasmacytoid dendritic cells. And mature PDCs means that they acquire the capacity to produce type 1 interferon and high amounts of type 1 interferon. This is like per cell, the amount of type 1 interferon per cell, and which was equivalent to ex vivo isolated PDCs. And they also acquired the morphology of mature cells as similar to what we identify when we sort splenic PDCs. So what we could establish was that about 80 to 90% of PDCs was generated from a lymphoid progenitor which expressed IL-7 receptor, and about 20% of PDCs was generated from myeloid progenitor. Their differentiation went through a committed precursor which expressed Siglec-H and Ly6D, and what I'm not showing here is that the expression of the TCF4, which is required to have or to generate this PDC identity is established only upon the acquisition of this committed precursor stage. What we observed was that the level of IRF8 or EBF1, which is required for B cell, was somehow determining the commitment towards PDCs or towards B cells within this lymphoid progenitor. And this is shown here when we compared the expression levels using a dual reporter mouse for IRF8 and EBF1. The single positive cells, which were mostly committed to the B cell lineage, expressed high levels of EBF1, while the Siglec H-Ly6D committed pre-PDCs expressed high levels of IRF8 and no EBF1. So what remained to be addressed was whether the differentiation from either subset, progenitor subset, was resulting in a unique cell, or whether each cell, depending on their lineage, would generate mature cells with different functional properties and identities. And in order to address this question, we performed single-cell RNA sequencing. So we sorted splenic PDCs from, or bone marrow PDCs, here in blue and in red, and looked at their expression profiles. So all cells shared a common PDC signature, which is characterized by their TCF4 or E22, the PDC identity transcription factor by IRF8, and by BSC2. However, when we looked at lymphoid genes or myeloid genes, we could identify two different clusters. One which was characterized by lymphoid transcripts and accounted for about 80% of the cells, 80 to 90%, corresponding to the differentiation data we obtained in vitro, and a small fraction of cells of about 10 to 20%, which was characterized by myeloid transcripts, such as LGALS3 or galectin-3, the transcription factor ZBTB46, which characterizes dendritic cells, and monocyte or macrophage genes such as CD14 and CX3CR1. So using the reporter mouse line for the transcription factor ZBTB46 in combination with BSC2 or CD317, we were able to segregate and sort those different subsets. And we decided to call PDCs, or the lymphoid cells PDC, conventional PDCs, and the cells which were characterized by this myeloid signature as PDC-like cells, given their similarities with PDCs. In terms of functional properties, what appeared obvious was that both subsets, independently of whether they had myeloid or lymphoid genes, they were both capable of producing type 1 interferon upon stimulation with CpG, suggesting that they share this capacity of antiviral properties among them. However, when we looked at the antigen presentation capacity, what we observed was that while conventional PDCs, or PDCs which had this lymphoid nature, were incapable of capturing the OVA protein, processing it, and presenting it to T cells to induce T cell proliferation, these myeloid PDCs or PDC-like cells were efficient in terms of promoting T cell proliferation to a similar extent, a bit less maybe, than conventional dendritic cells. So when we go back to the original hypothesis, it appeared that both cells were capable of producing type 1 interferon, but now the question was like, what about the myeloid properties which were identified or suggested, or which remain controversial within the PDC field, if those were related rather to this PDC-like subset instead of conventional PDCs? And so we decided to characterize more in detail these cells with a particular focus on these myeloid PDCs or PDC-like cells. And so this is again the gating strategy, showing conventional PDCs, PDC-like cells, and conventional dendritic cells, which either express SERP-alpha and are referred as CDC2, or express CXCR1 and are referred as CDC1. And what we observed was that those myeloid PDCs were very different from conventional PDCs based on their morphology. And they had this polylobated nuclei, which was reminiscent of some of the type 2 dendritic cells, which remain a very heterogeneous subset. So in terms of tissue distribution, PDC-like cells, which are here projected in orange, appear to be abundant in certain tissues, which so here in the heart, in the brain, or in brain and meninges, within the thymus or the bone marrow, but not so abundant in secondary lymphoid organs or in peripheral tissues such as the lung, suggesting that those tissues are, so within those tissues, we know that we rather want to prevent an immune response rather than generate an immune response, while in peripheral tissues, we need to really perform immunity. And what was important was that based on their profile, a corresponding subset of PDC-like cells that was referred as ASDCs or pre-PDCs, pre-DCs, had been identified a few years back in human peripheral blood. So we performed a legend screen analysis using 255 surface markers, trying to understand what was the overall identity of the cells. And in collaboration with Florent Ginoux, who's shared with us the pipeline, which is called Infinity Pipeline, we were able to look at all those markers within, as it was a like a site of experiment. And so as you can see here in this projection, PDC-like cells appear to be somehow an intermediate stage between conventional PDCs and type 2 dendritic cells. They express lower levels of Siglec-H, lower levels of BST2, but they also express high levels comparable to conventional DCs, which are shown here as DC1 or DC2, of the transcription factor ZBTB46. When we looked at the most differentially expressed markers, what we identified were a lot of markers which were in common with monocytes, such as CX3CR1 and CD14. However, none of the markers that we looked at appear to be unique to PDC-like cells. Most of them were either shared with PDCs or with either DC1 subsets, such as CD49E or CX3CR1. So we wanted to validate or to confirm, independently of the expression of ZBTB46, the fact were related to the DC lineage, or understand whether they were more related to the monocyte lineage. And here, this is based on the expression of ZBTB46. They appear to be more linked to conventional dendritic cells, and they express the marker CD26, which allows discrimination from monocytes. The other feature that defines the dendritic cell is definitely their dependency on FLT3 ligand. And so we looked at FLT3 ligand deficient mice, and we observed across all dendritic cell subsets, including PDC-like cells, a profound reduction in the absence of this cytokine, suggesting that they depend on FLT3 ligand and that they depend or that they belong to the dendritic cell lineage. So we then performed bulk RNA sequencing, and here you can see across different tissues how they cluster based on a principal component analysis closer to plasmacytoid dendritic cells, and then within the DC compartment closer to type 2 dendritic cells rather than type 1 dendritic cells. In terms of like general transcript, looking at surface marker or transcription factors, they appear indeed much more similar to plasmacytoid dendritic cells rather than conventional DCs. And within the conventional DCs, they appear to have most in common with thymic derived type 2 dendritic cells. In terms of transcription factor, they indeed express TCF4, so the PDC transcription factor, characterizing transcription factor, but also KLF4 or Cooper-like factor 4, which was shown to be important for a specific subset of type 2 dendritic cells. So when we performed pairwise comparison, looking at the transcript between PDCs and PDC-like cells, what we observed was that PDC-like cells were characterized by a strong dendritic cell signature. On the other side, when we compare PDC-like cells with conventional DCs, PDC-like cells had a strong plasmacytoid dendritic cell signature, again, confirming or suggesting this intermediate phenotype and this intermediate transcriptional profile between PDCs and dendritic cells. When we performed pathway analysis, one thing that appeared obvious, besides this enrichment in inflammatory response, was the fact that they had a lot of genes which were identifying potentially proliferation. And so we decided to look whether those cells had proliferative capacity. So this is conventional PDCs, and conventional PDCs, independently on which cytokine or which culture condition you put them, they are very fragile and they don't survive in vitro. However, when we looked at PDC-like cells, what we observed was that whenever we added homeostatic cytokines such as GM-CSF, IL-3 or MCSF, PDC-like cells proliferated significantly. In terms of, so we then transferred those cells in vivo, and when we looked using the CD45.2 congenic marker, what we saw was that the cells that looked like PDCs acquired a type 2 dendritic cell. So they induced high expression of MHC class 2, high expression of CD11C, but also SERP alpha, suggesting that they could be precursor, that they were precursor of type 2 dendritic cells. And here is quantified the percentage of output in terms of different subset, while conventional PDCs showed almost no plasticity. We knew that the cells were expressed the transcription factor KLF4, and so we had the mice, so we looked at KLF4 conditional knockout mice, and what was known was that there was a reduction in the type 2 dendritic cell subset. When we looked at PDC-like cells, instead of a reduction, we observed an increase, and so we reasoned that potentially in KLF4 deficient mice, there was a differentiation block which prevented the differentiation into type 2 dendritic cells, which resulted in a decrease in the DC2 subset. So to look more formally into that, we transferred again in competitive setting PDC-like cells from either wild type or KLF4 deficient mice and looked at their output. And in terms of engrafted cells, there was not a big difference, suggesting that both cells were efficiently capable of giving rise to progeny. However, the KLF4 deficient cells were very poor in terms of generating type 2 dendritic cells with a significant reduction of about half of the cells, and they remained mostly PDC-like cells. Some of the cells deflected into conventional PDCs or type 1 DCs. When we looked in terms of like type 2 dendritic cells, the one subset which was mostly affected by the deletion of the transcription factor KLF4 was the one which expressed CX3CR1, which was recently referred by Brown as CDC2B. So we then performed single-cell RNA sequencing, and here are the different clusters that we obtained. And we did that combining wild-type or KLF4 deficient mice. And here you can see the type 1 dendritic cells, conventional PDCs, type 2 dendritic cells, migratory disease. And what was striking within these PDC-like cells was that the subset appeared to be split, with some cells in close proximity with the CDC2, and in particular the CDC2B, and some cells which clustered in proximity with conventional disease. When we split into the two genotypes, the conventional, so PDC-like cells from wild-type mice resembled or like had a profile which was much closer to type 2 dendritic cells, while PDC-like cells from the knockout mouse remained in proximity to PDC, suggesting that they remained within this PDC type of enriched transcript. And we saw a profound reduction in the type 2 dendritic cell subset, and in particular in the subset which expressed CX3CR1. And here you can see the subset projection for the expression of CX3CR1 or CLECTN-A, which was suggested to be the CDC2B. So when we put this together, okay, let me close this. When we put this together, what we were able to show was really that most PDCs or conventional PDCs are derived from a lymphoid progenitor. They go through a precursor stage, which is expressing Ly6D and Siglec-H, which is committed, and acquire at this point high levels of TCF4. And they have limited, if no, antigen presentation capacity. On the other side, these PDC-like cells appear to be a circulating progenitor subset for type 2 dendritic cells, and in particular for CX3CR1 CDC2B. This differentiation was dependent on the transcription factor KLF4, and this subset was the one which was capable of performing antigen presentation. Oops. Okay. So what about the mature cells? How many type 2 dendritic cells derive from this PDC-like progenitor? And to address this question, we took advantage of the reporter line or the lineage tracing line, which has the human CD2 Cre crossed with the ROSA 26YFP. So this line, only the lymphoid compartment is labeled, and therefore also PDCs and PDC-like cells are labeled. When we looked at the number of type 2 dendritic cells which are labeled, we end up with 25%, suggesting that at least 25% of type 2 dendritic cells appear to derive from these PDC-like cells. It could be more because some of the cells, some of the PDC-like cells are not labeled. But what we can say for sure is that at least 25% of DC2, and in particular CDC2B, appears to derive from this circulating progenitor. But what about their function? We knew that they were capable of performing antigen presentation, and so we tried to address more in details what type of the response and when they would be responsible for driving T cell proliferation and activation of adaptive immunity. And so when we stimulated with different TLR agonists, what we observed, this is like the column in blue, was that PDC-like cells appear to be as competent as conventional DCs to induce T cell proliferation. And in some circumstances, they were even better than conventional DCs. The fact that they were better was, however, linked to their progenitor nature, as when we looked at the number of dendritic cells which were still in culture at day 3, this was mostly dendritic cells. This was mostly within the PDC-like cells, suggesting that the fact that they were capable of proliferating and differentiating into dendritic cells most likely allowed for more T cells to be stimulated by each PDC-like cells as compared to conventional dendritic cells. So when we looked at KLF4 deficiency, what we observed was that this abrogated completely the proliferation capacity, suggesting that this differentiation into what is a conventional DC or what looks like a conventional type 2 dendritic cell or CDC2B was the reason for their antigen presentation capacity. We also looked at IR4 conditional knockout mice, and in vitro there is no difference in terms of like antigen presentation, suggesting that this capacity of PDC-like cells to drive T cell proliferation is really dependent on their capacity, on the drive that KLF4 allows to differentiate into conventional DCs, and in particular CDC2B. In terms of cytokine production, the PDC-like cells were producing high amounts of the IL-6 interleukin, IL-22, as well as IL-10 and IL-17. And this is comparing conventional PDC-like cells or conventional DCs. And all those cytokines are indicative of a pro-inflammatory or autoimmune type of phenotype. But the fact that they produce also IL-22 suggests, in combination with IL-17 and IL-10, suggests that those could also be related to tissue repair functions. The capacity of T cells to produce IL-17 was again abrogated in the context of KLF4 deficiency, suggesting that both the proliferation, the activation, but as well as, but also the polarization into Th17 producing cells was abrogated in the absence of this differentiation into conventional dendritic cells. And we then tried different types of stimulation, which lead to IL-17 polarization. But what we observed was that under steady state conditions, we already have a reduction in the CD4 IL-17 positive T cell pool. And this was true for the skin draining lymph node as well as for the skin. So suggesting that within the epithelial tissues, the lack of this transcription factor, which prevents the differentiation of PDC-like cells into conventional type 2 dendritic cells, leads to a reduction of the resident pool of IL-17 cells. So with this homeostatic reduction. So let me go back to the final model. Like, so here is like the PDC, which derive from lymphoid progenitor, and the PDC-like cells, which really seem to be a pool of cells capable of differentiating into effective antigen-presenting cell under homeostatic cytokines. So in terms of response, the one thing that we are now hypothesizing, and we have some very preliminary evidence, but we need to confirm it, seems that the response of this Th17 cells seems to be related to the microbiome. And we are trying to understand how much this could be influencing the immune response towards self, and could potentially lead to the development of autoimmune syndrome, such as lupus or Sjogren's syndrome. But they seem really to control this homeostatic pool, which is known to be dependent on the microbiome. And so with that, I would like to acknowledge all the people who contributed to the research, and there are for sure more people which are not listed here, but in particular Patrick, who did most of the work, which I presented to you, between the University of Basel and the NIH during a pandemic. So it was quite a big challenge. All the people from Basel and from the NIH, the genomic core facility of the university in Zurich, and the bioinformatician, here Robert Ivanek, who performed the study, collaborators across the world, in particular Florent Ginou, who is now located in different places, collaborators in WashU, in particular Marco Colonna, who helped us with generating a reporter line, the collaborators within the NIH, and the funding agency, initially in Basel and now currently at the NIH. And thank you all for the attention.